Assalamu alaikum, dear students. Welcome to the course Principles in Animal Life 1. This is lecture number 54, and in this lecture we will start discussing various cell organelles. The first two are nucleus and ribosomes. Why we are discussing these two organelles together? Because they are related with each other functionally. Nucleus carries genetic information, and for the expression of this genetic information, the services of ribosomes are required, because ribosome is the site where the translation of messenger RNA takes place. Discussing agenda for this topic will be: the eukaryotic cells' genetic instructions are housed in the nucleus and carried out by the ribosomes. So messenger RNA is copied from a gene through the process of transcription, and then this messenger RNA settles on the surface of ribosome, where the ribosome, in association with transfer RNA, decodes the genetic information it has and synthesizes a protein or a polypeptide chain via a process called translation. The nucleus, which is of course the information center of the cell, or the control center of the cell, and then we will discuss ribosomes, which are the protein factories, protein workbenches, universal organelles. Lecture outcome. After watching and listening this lecture, students will know about the nucleus, its structure, function, and its association with ribosomes in the expression of genetic information it contains. So the eukaryotic cell's genetic instructions are housed in the nucleus and carried out by the ribosomes. In a eukaryotic cell, the nucleus, which is a distinct organelle, resides in the center of the cell or near the center, that is, in the animal cells. In plant cells, because there is a large vacuole which occupy most of the area of the cell, so the nucleus in plant cells is pushed towards one side of the cell, but in plant cell also it is a distinct organelle, and it is of utmost importance for the cell, because it is the control center of the cell. It houses the genetic material, it houses the DNA or the chromosomes. Now these two cellular structures, they are very important. They are functionally related with one another. The nucleus houses the DNA, it stores the genetic information, and the ribosomes, which uses information from DNA, that is, the messenger RNA which is copied from DNA, finds itself on the ribosomes, and then ribosomes, in association with transfer RNA, synthesizes a protein, a polypeptide chain from the instructions present in the messenger RNA. So these two organelles are related with each other. If you remember the central dogma of life, or the central dogma of molecular biology, which says that the transfer of genetic information in a biological system occurs from DNA to RNA to proteins. So DNA carry genetic instructions, and these genetic instructions are copied to messenger RNA, and then messenger RNA finds itself on the ribosomes, where a polypeptide chain or a protein is synthesized. So the nucleus contains most of the genes in the eukaryotic cell. Why they are saying most of the genes? Because in eukaryotic cells, the mitochondria or the chloroplast also have their own genetic material. So some of the genes are also present in chloroplast and mitochondria, but majority of the genes reside in the nucleus in eukaryotic cells. Now, it is usually the most conspicuous, most distinct organelle, and its size is 5 micrometer in diameter. Now the nuclear envelope encloses the nucleus. Now remember that inside eukaryotic cells, there are two organelles which are bounded by double membrane. That is two membranes. One of them is nucleus and the other is mitochondria. Both these organelles are bounded by two membranes, or they are double membraned. So the nucleus has a double membrane structure around it, and it separates the nucleus from the cytoplasm. And remember from our last lecture, we said that the composition of the membranes surrounding different organelles is different. Why it is different? Because it is supplied with such structures which helps in carrying out the functions that particular organelle is reserved for. For example, mitochondria has complexes embedded in the internal membrane which has electron carriers. The nuclear envelope is a double membrane. So here you can see this is the nucleus, and within the nucleus there is a structure called nucleolus. I hope you remember the function of this structure. This is the place where the ribosomes are synthesized. Ribosomes are made up of equal amount of ribosomal RNA and proteins, and these two components are assembled in nucleolus. And then you have this chromatin. Chromatin is composed of DNA and associated proteins. Associated proteins may be histones and non-histones, and then these are the two membranes, because nucleus is surrounded by two membranes, so the outer membrane and the inner membrane, and then we have nuclear pores. It is of course for the transport of materials between the cytoplasm and the nucleus, but these nuclear pores are supplied with proteins. So this whole structure, this nuclear pore surrounded by a set of proteins, it form pore complex. And chromatin, as already said, is the combination of DNA and associated proteins. Now this is the structure of DNA and associated proteins, that is histones. So this is a histone octamer. There are five different types of histones: H1, H2, H1A, H2B, H3, and H4 and H5. So these five types of histones, they are in association with DNA. Four types of histones: H2A, H2B, H3, and H4. These are called core histones because they are present in this nucleosome and the DNA is wrapped around them. And then we have a fifth histone, which is H1 or H5, and it is present here in the middle between the two nucleosomes, and it is also called linker DNA. And here you see these structures, these are the histone tails. Histones are of course proteins and they are made up of polypeptide chains. So these are the histone tails, and the process of acetylation and methylation takes place on these tails. So the nucleus houses the genetic material, and the genetic material or DNA is in association with histones. So these histones helps in the condensation of the genetic material. If you remember, the amount of DNA or the length of DNA inside a single eukaryotic cell is 1.5 to 2 meters, and this large molecule of DNA is assembled in a place which is only 5 micrometer in size. So just imagine that the nucleus is housing a structure which is 250,000 times greater than its own size. And it is only possible when this DNA condenses. When it condenses 10,000 times, then it is able to accommodate in a space of 5 micrometers. So there are different degrees, different levels of condensation. There is coiling and then supercoiling and then yet another level of coiling where this process occurs, and only then is DNA is in a position to accommodate inside the nucleus. The two membranes, each a lipid bilayer with associated proteins, are separated by a space of 20 to 40 nanometer. So there is a space of 20 to 40 nanometer between the two membranes. The envelope is perforated by pore structures that are about 100 nanometer in diameter. So this nuclear envelope is perforated. There are pores at various locations in this membrane, and these pores are for the transport of materials. For example, the transport of larger and smaller subunit when it is synthesized on nucleolus to the cytoplasm, the transport of the messenger RNA or other types of RNAs to the cytoplasm from the nucleus. So this nuclear pore provides a passage to those materials, and the diameter of nuclear pore is 100 nanometer. At the lip of each pore, the inner and outer membranes of the nuclear envelope are continuous. So at the point where the pores are present, both the membranes, inner and outer membrane, they are continuous with one another. So suppose this is one membrane, this is the another, and Here they have a nuclear pore. So here at that point the inner and outer membrane will be continuous with one another. This is the nuclear pore. And this nuclear pore is surrounded by proteins which forms a pore complex. Now, at an intricate protein structure called a pore complex lines each pore and plays an important role in the cell by regulating the entry. So, you can say that these proteins are the selective gatekeepers which regulate the movement of molecules between the plasma membrane, between the cytoplasm and the nucleus. Except at the pores, the nuclear side of the envelope is lined by the nuclear lamina, a net-like array of protein filaments that maintains the shape of the nucleus by mechanically supporting the nuclear envelope. So the nuclear lamina is a structure which is composed of cytoskeletal elements and these cytoskeletal elements enables the nucleus to stay in a proper shape. There is also much evidence for a nuclear matrix, a framework of protein fibers extending throughout the nuclear interior. The nuclear lamina and matrix may help organize the genetic material so it functions efficiently, so it not only provides a proper shape to the nucleus but it also helps in the proper organization of the genetic material. Means it these are a kind of scaffolding structures for the genetic material. Now within the nucleus, the DNA is organized into discrete units called chromosomes. So chromosomes are basically composed of DNA and associated proteins. The associated proteins may be histones or non-histone proteins. Now remember that the chromatin also has the same composition, DNA plus associated proteins, and the associated proteins are histones or non-histones. And what is the difference between the chromosomes and chromatin? They have the same composition. The difference is the degree of condensation. So chromosome is the highly condensed form of chromatin, and remember chromosomes are only visible during cell division, and chromosomes are the vehicles which carry the genes, the functional units of heredity. Each chromosome consists of one large molecule of DNA, and we have already discussed about histones which are directly associated with DNA. 5 types of histones: H2A, H2B, H3, H4 and H5. The 4 histones which are present in a pair form, and they form the histone octamer. Histone plus DNA forms the nucleosome, and histones are supplied to the DNA for 2 reasons. First, they helps in the regulation of gene expression, and secondly, it helps in the condensation of the genetic material so that it can accommodate within the nucleus. Now, some of the proteins help coil the DNA molecule of each chromosome, reducing its length and allowing it to fit into the nucleus. We have already discussed that. Now, the complex of DNA and associated proteins making up chromosome is called chromatin. So basically they have the same composition. The only difference is the degree of condensation. And remember that chromatin is the form where the genetic material is present during interphase. I hope you remember interphase, which is the G1, S and G2 stages. So this period is called interphase, and it covers 90% of the time of the cell cycle. So during interphase, the chromosomes are not visible. The chromatin is present in a highly diffused form, and there are two distinct regions within the chromatin. One is called heterochromatin, which is a region where the DNA and histones have tight association, and this is a region which is gene deficient, or where the genes are not expressed quite often, and this is the region which is darkly stained, whereas the other region, which is called euchromatin, is usually a region which consists of housekeeping genes, or the genes which are quite often expressed. The euchromatin is the transcriptionally active area of the genome, and it is the area which is lightly stained. So in the interphase, if you apply certain coloring agents, certain dyes to chromatin, then you will see these two distinct regions, euchromatin and heterochromatin. Now, as the cell prepares to divide, there is considerable amount of coiling and condensation. Remember that the 1.5 to 2 meter DNA will have to condense 10,000 times to accommodate in a space of 5 micrometer diameter. So each eukaryotic species has a characteristic number of chromosomes. Now the number of chromosomes vary from species to species, but remember one very important sentence, that the chromosome number in a species does not depend upon the size and structural complexity of that species. So we cannot say that a mice will have lesser number of chromosomes and an elephant will have large number of chromosomes because of the size difference, or a Drosophila will have lesser number of chromosomes because it is an invertebrate, and a complex vertebrate will have high number of chromosomes. No. Once again repeating, the number of chromosomes in a species does not depend upon the size and structural complexity of that species. So, for example, humans have 46 chromosomes, and the primates or the monkeys, they have 48 chromosomes. The potato also has 48 chromosomes, and then you have pigeon, which has 78 chromosomes. Drosophila has 4 pairs of chromosomes. So the chromosome number varies between individual, and there are even microscopic organisms which have 1200 chromosomes. For example, a typical human cell has 46 chromosomes in a nucleus, and the germ cells have half number of chromosomes as that of the somatic cells. The fruit fly has 8 chromosomes which are arranged in 4 pairs, and there are of course 4 chromosomes in the germ cells. The germ cells carry half the number of chromosomes as that of the somatic cells. Now, a prominent structure within the non-dividing nucleus is the nucleolus. The nucleolus appears through the electron microscope as a mass of densely stained granules and fibers, adjoining part of the chromatin, and it is the site where the ribosomal RNA is synthesized from genes in the DNA, and then the proteins are imported from the cytoplasm through nuclear pores to the nucleus, and the smaller and larger subunits of the ribosome are synthesized and assembled. So the large and smaller subunits, the assembly is basically between the ribosomal RNA and the associated proteins. So larger subunit and smaller subunit, both of them are synthesized inside the nucleolus, and then they are transported out to the cytoplasm, where the small and large subunit assemble with each other and form the ribosome. Sometimes there are two or more nucleoli. The number depends on the species and the stage in the cell's reproductive cycle. The nucleoli may also play a role in controlling cell division and the lifespan of a cell. So now we are moving towards the ribosomes. The nucleus directs protein synthesis by synthesizing messenger RNA, and then this messenger RNA travels to the cytoplasm, that is in case of eukaryotic cells, where it is translated. So the messenger RNA is then transported to the cytoplasm via nuclear pores, and here it gets translated, and a polypeptide chain is synthesized. Now, before discussing ribosomes, one sentence to summarize the nucleus. So the nucleus resides in the center of the cell. It is a conspicuous, distinct organelle. It is the information center of the cell. It is the control center of the cell. And what are the functions of the nucleus? So the nucleus is responsible for the protection, expression, and replication of the genetic material. Now, ribosomes are the protein factories. They are the protein workbenches. They are the universal organelles, and these are involved in the process of translation. Ribosomes are made up of an equal amount of ribosomal RNA and proteins, and ribosomes are not considered as organelles because they are not bound by any membrane. So that explains the reason why they are present both in prokaryotes and eukaryotes, because they are not bounded by any membrane. Remember that eukaryotes have membrane bounded organelles and prokaryotes lack membrane bounded organelles. But this ribosome is an exception. It doesn't have any membrane at all, so it is present both in prokaryotes and eukaryotes, and it is a universal organelle because it is present in almost every type of cell. Because its function is important, it is responsible for the process of translation. The process of translation takes place on the surface of ribosome in association with transfer RNA. So it is a translator or interpreter for the nuclear information, and it translates the nuclear information into a language of proteins. Now we are going to discuss several examples of that important phrase in biology which says that structure determines the function. So the cells which are actively involved in the synthesis of proteins will have large number of ribosomes. And for example, human pancreatic cells which actively release digestive juices, they have millions of ribosomes. Now where the ribosomes are found, there are two types of ribosomes in a cell. In a eukaryotic cell, one type is the free ribosomes which are suspended in the cytosol, and the other are bound ribosomes which are attached with the endoplasmic reticulum. And because of these attachment of bound ribosomes, the endoplasmic reticulum appears rough, so it is called rough endoplasmic reticulum. Here you have the two subunits of the ribosome. The larger subunit is the site where actually the process of translation takes place. The large subunit has three distinct locations: exit site, E, peptidal site, and then the amino acyl site. So there are three distinct sites in the large subunit where translation takes place. Now both bound and free ribosomes are structurally identical. There is no structural difference between them, and they can also shift their positions. For example, a free ribosome may get attached with the endoplasmic reticulum and becomes a bound ribosome, and vice versa. Most of the proteins made on free ribosomes function within the cytosol. Examples are enzymes that catalyze the first step of sugar breakdown. Now remember that the ribosomes are concerned with the translation of messenger RNA into a polypeptide chain. So this process can occur both on the bound ribosomes and also on the free ribosomes. But remember one thing, that those proteins which are intended to stay within the cell and function within the cell, the RNAs of such proteins are translated on free ribosomes, whereas those proteins which are destined to be transported outside or to the membrane, the RNAs of those proteins are translated on the bound ribosomes. Now, cells that specializes in secretion, that is the cells which secrete proteins or digestive enzymes, they will have large number of bound ribosomes, because the proteins which are destined to leave the cell, their mRNAs are translated on bound ribosomes. And that is the end of the lecture. For details, you can refer to Campbell's book, and you can also ask questions on KCMS and WhatsApp groups. Thank you.